Corynebacterium Diphtheriae. That's the Chinese name, okay? Okay, here, the genus name Corynebacterium means that it's a club-shaped bacteria. So you can check the specific shape of the cell. This one with this one, you can see that on one end, it's wider than the other end. So makes the cell look like a club-shaped cell. So that's the name of Corynebacterium. And if you stain the bacteria with aniline, it's the chemical, and you would see some deep staining granule in the cell. And this deep staining granule in Corynebacterium diphtheriae is not endospore. It's just the inclusion bodies. Okay, the inclusion bodies which accumulate some material in it, the polyphosphate. Okay, and this can provide the bacteria with some energy once needed. And under the microscope, in the smear, the individual bacteria, the Corynebacterium, would lie in parallel. So the two different bacterial cells would lie in parallel, or they lie in acute angle, okay, a small angle, acute angle to each other. So that's the typical morphology under under light microscope. When we grow the diphtheria bacteria, they grow better in the presence of oxygen. They grow aerobically, and they are not picky to their medium. So they can grow in a lot of different type of media. Okay? They're not picky to their nutrients. If we incorporate a chemical called potassium tellurite, a special chemical, potassium tellurite, into the agar, when the bacteria grow on the surface, they would show this brown-black halo, brown-black colony with a halo. So this medium containing potassium tellurite is a differential medium, and if you see a colony like this, you would know that this is the diphtheria bacteria. And when we talk about the bacteriophages, we mentioned the term lysogenic conversion, and we mentioned that if the phage infects some bacteria, the phage genome might carry some virulence factor. And this is a famous example of this. The bacteriophage of diphtheria, the Corynebacterium diphtheriae, it actually carries the toxin gene on the phage genome. So when the phage infects this bacteria, it inserts its DNA, its genome, into the bacterial genome so that the bacteria would become toxigenic. It's able to produce exotoxin now. Okay, so without phage infection, it's not toxigenic. After infection, it's toxigenic. And this is a famous example of lysogenic conversion. We should recall something we learned before. Pathogenesis of diphtheria. It's spread by drops or by contact, droplets or aerosol, so that infected person, when they cough, they would release the bacteria to the air in droplets, and it can cause person-to-person spread. And all the toxigenic diphtheria are capable of elaborating diphtheria toxin. And this toxin is, okay, it's a two fragments toxin. It's an exotoxin with AB structure. While the A fragment is the active fragment, which can cause problem, while the B fragment is the binding ligand of receptor. So what happens is that when the bacteria produce the toxin, the diphtheria toxin, the B fragment bind to the receptor on the cell surface, then internalize the toxin into the cell. Then the A subunit is released. That's the toxic part of the toxin. A subunit inactivates the elongation factor 2. Elongation factor 2 is needed for protein synthesis, okay? So that the toxin can inhibit the protein synthesis in the cell. And finally, the cell will be killed because the cell cannot produce its own protein. It can be killed. So this is how the diphtheria toxins function in the human cells. And in the real world, the diphtheria, the toxigenic diphtheria can infect the little kids. And in the throat part, you would find a special structure in the kids. After infection, you would find a pseudomembrane there. The pseudomembrane contains the dead cell, the killed epithelial cell by the toxin. And also it contains fibrin protein. Last time we mentioned the fibrin protein. That's the insoluble protein network. And also you would find a lot of red blood cell and white blood cell in the pseudomembrane. And also a lot of dead epithelial cell, so that it forms a membrane in the throat part. That's why diphtheria infection is dangerous, because the pseudomembrane in the throat part can block the airway. Once the kid cannot get in enough oxygen, they will be killed. That's a major problem. So the kids, after infection, would have a sore throat, low-grade fever. Then they would suffer from prostration and dyspnea. So they cannot breathe well. They cannot get in enough oxygen. And if that develops, they would probably die due to suffocation because they cannot get oxygen. The patient also suffer from irregularity of cardiac rhythm, so the heart problem, swollen neck lines, and also difficulties with vision, speech, swallowing, or movement of the arm or legs. Okay, so here's an example of diphtheria infection in a 10-year-old boy. And you can see here the swelling of the neckline, okay? Okay. This is for the laboratory test. Like specimens, you check the smear, you will find a typical stent granules, and like culture uses selective medium like the telluride plate. I'll just leave this for you guys to learn. I would like to introduce a special test called ELEG test. ELEG test is designed to detect the presence of exotoxin produced by the toxigenic diphtheria. Let me explain how it works. Okay. Here, this is a round one. This is the culture plate, the agar plate. We culture the bacteria. So before we pour the medium into the plate, we put a strip, a paper strip, this one. The paper strip at the bottom of the plate. on the strip, okay, it contains the antitoxin, the antibody on the paper strip. Okay, put it on the bottom, then you pour the agar on top of the paper strip. Okay, after it's solidified, it cools down, then you streak the bacteria on the surface to cross the paper strip. Okay, when you culture the bacteria on the surface, you culture the known strain which can produce toxin. This is a known toxigenic bacteria, okay, in the line. You also culture a known strain which cannot produce toxin. Then you streak a line of your patient's isolate. When you culture the bacteria on the surface, the bacteria is able, if it's toxigenic, it's able to produce toxin. The toxin would diffuse into the agar, into the plate, okay? It diffuses into the agar, and meanwhile, the antibody in the paper strip also diffuse into the agar. When the antigen and the antibody meet, they would interact with each other so that you would see a precipitating line like this. You can actually see it. This is a real plate. You can see the precipitating line. If you see the precipitating line, you would say that this strain is toxigenic, while this strain is not toxigenic. And this is the unknown patient's isolate, and you can also see the precipitating line so that you know this patient's isolate is toxigenic. Okay? So that's the ELIC test. You want to understand how it works. And if you have some question on this test, you would explain, you would be able to interpret the results. Okay. To treat the diphtheria infection, we can use the antimicrobial drugs like penicillin and other antibiotics. But for the active patient, they are already infected, you can provide immediate protection with antitoxin. So we mentioned before that antitoxin is the antibody which can neutralize the toxin. So this is the work happened over 100 years ago. Scientists use a small amount of toxin or toxoid to immunize the horse, okay? To immunize the horse. Then they would be able to harvest the blood, harvest the serum, which contains the antitoxin. And they can inject this antitoxin to the infected kids so that the antibody in the antiserum can provide an immediate protection, neutralize the exotoxin so that the kids can be rescued. So with this method, a lot of kids are rescued, survived. So this is after infection, you can cure the patients. This work was done by Dr. Emil van Buren. Emil van Buren. I think he's German, right? German, yeah. And so that's the first discovery of antibody which can neutralize a toxin. So they call it antitoxin. And Dr. Buren received the first Nobel Prize in physiology or medicine. So that's the first Nobel Prize in medicine. Very fantastic work. That's the time before people know anything about immunology, they discovered this useful molecule. to prevent diphtheria infection, I guess everyone in this classroom, you received the vaccine, the toxoid. We utilize a diphtheria toxoid. So we purify the toxin and then treat it with formaldehyde to inactivate the toxicity, but we can use it as a vaccine. So usually in China, the kids would receive a vaccine called DPT. Here, D stands for diphtheria toxoid, while T stands for tetanus toxoid. P means pertussis vaccine. It's inactivated pertussis. Do you have any idea whether you received this or not? A similar thing? No? I guess you received the similar thing. Diphtheria. Okay. And the patients with diphtheria infection should be isolated because there is person-to-person spreading. We can isolate the person till the person is recovered. Okay, another test we can run is a shake test. This is to test whether in a person whether you have the antitoxin, the neutralizing antibody in your body. So it's also a skin test. We just inject a very tiny amount, a minute amount of diphtheria toxin under the skin. Okay, under the skin. And this toxin, we know that it can cause cell death, right? And if your body does not contain any antitoxin, so if in your body you don't have antitoxin, you are not immunized, the toxin injected underneath the skin would cause cell damage, right? And it can cause cell death. It will cause inflammation surrounding the injection site. So in this person, it means that this person does not have any antibody. This person is not immunized, is not protected. While in this person, after injection, nothing happened at the site of injection, which means that the toxin injected in this site is neutralized by antibody, which means that this person is immunized and this person is protected. So with this Shakes test, we can check the whole population, like how many percent of the whole population is immunized, is protected. Very tiny amount of toxin underneath the skin. Okay, that's all for the diphtheria. Yeah, so that's all for today.